I guess.
Is it for the amount of time? Yeah.
I'm assuming.
From outside.
Okay, I'll make a beeping noise or something.
All right. Well, welcome to day number 2, and I hope everyone's enjoying Pittsburgh. A few housekeeping announcements. As people who've come for Pancreas Fest for many years, many of you're aware that during the pancreatic cancer session, the pediatric group, since it's not a problem for them, will split off. They're meeting in the lower lobby, which is sort of where the food is. There's a room right off of that. So when we get done with the announcements, if you're part of the pediatric group, that's where you should be. We'll reconvene at 10:15 for the after that break for the Ruth Bretsky Award and the rest of the sessions. A couple things. Please keep an eye on your email for CME instructions. That'll be coming out. There will be recordings of Pancreas Fest 2023 available for the next 90 days. And you'll so be aware of that. That's been asked. Also keep in mind that next year's Pancreas Fest dates will be in Cincinnati, July 25th and 26th. There will be an NIDDK workshop the day before on July 24th, and potentially other activities afterwards to be seen. Myson wanted me to make an announcement that her and Andrew Trout over the lunch hour will be having a breakout in the same room where the pediatric group is, and for everyone's welcome to attend. It's working on imaging guidelines for pancreatitis. There's trying to come out with consensus guidelines. So if you just want to get your lunch and go into that room, if you want to participate, it's open to all. I wanted to give a quick shout out. We couldn't do this meeting without the help of our industry partners. I wanted to highlight our gold supporters, the Nestle Health Sciences and Vivus, our silver with AbbVie, Cincinnati Children's Hospital and Medical Center, Digestive Care Incorporated, and Immunovia Incorporated. And our bronze sponsors, a Nationwide Children's Hospital, the University of Pittsburgh, and our poster sponsor was the National Pancreas Foundation. So thank you and all of our exhibitors for their support. Please, if you have a chance, stop by during the breaks, and good flow helps us keep the meeting going every year. And so with that, as you know, Myson's a stickler for keeping on time. So I will stop here, hand the meeting over to our moderators to take over for the first session. Thank you.
Morning. Myson, stickler for timing, also asked us not to, go over the whole history and credentials of the speakers. That information can be found online. So for this first session, Advances in Basic Sciences in Pancreatic Cancer, I'd like to introduce Dr. Channing Dare, the Sarah Graham Keenan Distinguished Professor at UNC Chapel Hill at the Lineberger Comprehensive Cancer Center, talking about targeting KRAS for pancreatic cancer treatment.
Thank you, and thanks to the organizers for the invitation to this meeting. Okay, so what I'd like to do is give you an update on the status of targeting KRAS for pancreatic cancer treatment. Helps to be on. Okay. All right. So first of all, disclosures. I have, in particular for this presentation, I do get financial support from Mirati Therapeutics as well as Revolution Medicines. Okay, so this is a word cloud of the genetic background of pancreatic cancer. And what should be very obvious to you is the dominant feature of this genetic profile where KRAS is dominating the landscape, where nearly 100% of pancreatic cancers have a KRAS mutation. And we know that KRAS is an initiating event for pancreatic cancer. And furthermore, that in mouse model analysis, that if you ablate KRAS expression, you can dramatically collapse pancreatic cancer. So it is in some ways an ideal target for pancreatic cancer treatment because of the addiction that pancreatic cancer has to a KRAS mutation. Okay, so because of these facts, targeting KRAS has been identified as one of the 4 key directions that the field has decided are priorities moving forward for advancing the treatment of pancreatic cancer. However, this has been easier said than done. When one looks at RAS, RAS has received the reputation as being an undruggable target. And this reputation comes from the fact that when you look at RAS as a structure, as my colleague Adrian Cox said at UNC, RAS is like a greasy ball. It seems like there's nothing to really grab hold of it with small molecule inhibitors. Frank McCormick, a leader in the RAS field, said a similar situation where RAS is like a tennis ball. If you can't find anything to attach it to, how are you going to target it? So, for better or worse, I've been involved in RAS research since the beginning, back in 1982. And it has been decades of frustration and disappointment. We have failed in many efforts to target RAS. However, a major breakthrough occurred in 2013 when Kayvon Shokat and colleagues developed the first direct RAS inhibitor. And in the process of identifying this RAS inhibitor, they identified a pocket that was not previously appreciated in RAS. And that pocket suggested that RAS indeed might be druggable. And in a remarkable 8 years from that initial discovery, we have the first approval of a KRAS inhibitor for the treatment of cancer in 2021. And so we now have 2 KRAS inhibitors that have been approved by the FDA in 2021 and 2022, sotorasib and adagrasib. And these are specific KRAS inhibitors for one specific mutation, the G12C mutation in lung cancer. And so, while this is great for lung cancer, unfortunately for pancreatic cancer, this mutation, which is a smoking-associated mutation, is seen at a less than 2% frequency. So, despite that low frequency, clinical trial analysis has shown that if you're a rare pancreatic cancer patient that has a G12C mutation, you do have a reasonably good response to a G12C inhibitor. So early clinical results suggest that these G12C inhibitors will also be responsive in the rare pancreatic cancer patients with the G12C mutation. Now, when you look at pancreatic cancer, the top 3 KRAS mutations are G12D, G12V, and G12R. So the task at hand is, can we develop inhibitors to target additional KRAS mutations. And what you can see on this slide here is indeed, there has been a lot of activity targeting G12C. In addition to the 2 approved inhibitors, there's a whole slew of additional G12C inhibitors that are now undergoing clinical evaluation. However, as I indicated to you, G12D is the major mutation, not only in pancreatic cancer, but in all cancers. So what is the progress in this area here? Well, progress has been actually quite impressive. And a number of G12D inhibitors are now under preclinical or clinical evaluation. So what I like to do is tell you a little bit about a few of these inhibitors just to give you a sense of where the activities are being seen. So first of all, this inhibitor from Mirati Therapeutics, MRTX1133. This is an inhibitor that's selective for the G12D mutation. And in the slide here, there are 2 things that we wanted to look at. First of all, how effective is this inhibitor against G12D versus other mutations? And on the left panel, what you can see is that indeed it potently suppresses KRAS signaling through the MAP kinase pathway in the presence of a G12D mutation. There's activity against other mutations as well, but these activities are not to the same degree as what you see with G12D. And on the right panel, what you can see is treatment of several G12D mutant pancreatic cancer cell lines. And what you can see is very impressive low nanomolar inhibition of the growth of KRAS G12D mutant pancreatic cancers. Moving on to a second inhibitor. This is one developed by Revolution Medicines. This one is distinctly very different. It works by a more complicated mechanism, a tri-complex mechanism. And furthermore, it involves the covalent modification of RAS. And what you can see here is on the left, there's an upper band that you can see, which represents the covalent modified form of RAS. And you can see that this version can be seen at low nanomolar levels. And on the right panel, this inhibitor was then used to treat a series of pancreatic cancer cell lines with G12D mutations. And what you can see is impressive low nanomolar potency in blocking the growth of KRAS G12D mutant cancers. Let me move on to a third inhibitor, which is one of the more provocative ones. I've told you about G12C selective inhibitors, and I've just told you about 2 G12D selective inhibitors. This one is very different. It's called a RAS motie inhibitor because rather than trying to target one RAS mutation, this inhibitor targets multiple RAS mutations. And in this panel here, what you can see here is that we evaluated the ability of this inhibitor to block signaling through the MAP kinase pathway with a whole range of different KRAS mutations. And the bottom line is that this inhibitor holds true to its name. It can indeed block the activity of multiple RAS mutations. And so these studies are now under review at Nature. And so we'll hopefully hear more about this in the coming weeks. So here is, we evaluated the activity of this inhibitor in a panel of KRAS G12D mutant pancreatic cancers. And what you can see is impressive single-digit nanomolar potency in blocking KRAS G12 signaling and the growth of KRAS G12D mutant cancers. And so there's yet a second manuscript under review at Nature that shows a comprehensive determination that this inhibitor can block pancreatic cancer growth in a wide range of pancreatic cancer models. And these are studies that are done with Ken Olive, Bob von der Heide, and Ben Stanger. Okay, so at this point, I've shown you some quite impressive development of RAS inhibitors against multiple KRAS mutations. And at this point, are we done? Well, unfortunately, no. This is a summary of the clinical trial results with the different G12C inhibitors against different cancers, lung, colorectal, and pancreatic cancer. There are several things here which cause us to pause and say, well, we're definitely not done yet. Objective response. Even though all these mutations have a G12C mutation, less than 50% of them respond initially to these inhibitors. And secondly, if you look at the duration of response, those that do respond, the response is short term. The average response rate is about 6 months. So these are the issues that we're facing now in the field. One is innate resistance. Why do cancers, even though they have the mutation, not respond to these inhibitors? Secondly, acquired resistance. Essentially, every patient that responds relapses and resistance arises. So these are some of the key issues that the field is now trying to address. And I'll briefly show you some of the analysis that we've done to try to begin to make some progress in these two areas. So several studies came out and described relapse patient mutations. And in these patients, mutations were found associated with relapse. And not surprisingly, most of these mutations involve components of the RAS signaling pathway. And in particular, most of the mutations seem to involve reactivation of RAS signaling. RAS signals through two key pathways, the ERK MAP kinase pathway and the PI3 kinase AKT mTOR pathway. So we asked the question, which of these pathways are the most important for driving resistance? We first looked at the MAP kinase pathway, and the bottom line here is that if you have mutations in the MAP kinase pathway, you can drive near complete resistance to these inhibitors. In contrast, when we did the other experiment to ask the importance of the PI3 kinase pathway, surprisingly, despite activation of this pathway, there was essentially no driving of resistance. Based on these findings, we said we must focus on the ERK pathway. So there actually are many inhibitors that have been developed against this pathway. That's the great news. Unfortunately, when taken to the clinic, these have not been successful. And to date, no inhibitor of this pathway has been approved for the treatment of any RAS mutant cancers. This is an example of the problem that we face. In some collaborative studies that I had with colleagues at Washington University, we had a clinical trial using an ERK inhibitor. And my clinical colleague, Andrew Wine-Galen, said this is one of the most difficult clinical trials that she has ever had to run, simply because patients just wanted off on the trial due to toxicity. So what is the solution to dealing with this toxicity issue? Well, there are two. The first involved looking for combinations to overcome the toxicity issues. And we've certainly taken this approach. We've identified a multitude of combinations to potentially offset the toxicity of an ERK inhibitor. I just want to talk about one. One was published in 2019 in Nature Medicine, targeting autophagy. So our study, together with two others, identified a combination of an autophagy inhibitor together with an inhibitor of the MAP kinase pathway as a promising therapy for pancreatic cancer. And in the second study by Martin McMahon and Colin Kinsey, they showed a proof of concept with one pancreatic cancer patient where this patient with a KRAS G12R mutation responded quite dramatically with a rapid tumor regression and basically loss of many of the tumors. Unfortunately, this patient relapsed and the tumors came back and the patient died of the cancer. So I want to emphasize this mutation because not all RAS mutations are the same. In a study that we published in Cancer Discovery in 2020, we looked at different RAS mutations and we concluded that this mutation is a little different than others. I won't go through the details except to say when you look at the right, we found that this mutation responded differently to different drugs compared to other RAS mutant pancreatic cancers. And the bottom line is that we think that this particular mutation may be more responsive to the combination of a MAP kinase inhibitor together with hydroxychloroquine. And I just want to tell you a story here. Last October, a patient reached out to me and said, I have a G12R mutation and I read your paper. Do you have any progress since the time of that publication? Is there anything that you can recommend to me for treatment? I suggested several options, but I also told him that Well, we did identify a possible combination, and there have been some exceptional responders. This is another option for you. I did not hear from this patient for months and months, but back this recent May, he contacted me and said he was finally able to get a hold of these inhibitors. He convinced his oncologist to use these inhibitors for compassionate use. And he said that his CA 19-9 levels plummeted and that scans showed that his tumors were regressing. And to the right, he provided the data from his analyses, and what you can see is a dramatic reduction in CA 19-9 levels. So this is a promising therapy, but what we don't know is why some patients respond and why most patients don't respond to this combination. So finally, the last thing we have done is to try to understand how ERK drives resistance to RAS inhibitors. And we've done a detailed molecular dissection of what ERK does. And I'll just give you a tip of the iceberg. ERK regulates a lot of things. It's a kinase, so it phosphorylates a lot of proteins, and many of those proteins are transcription factors. So ERK regulates a lot of gene expression changes. So we looked at the history of looking for KRAS-regulated genes, and we found many different studies. But you look at these studies, and there's actually shockingly very little overlap between each of these studies, which makes you wonder how sound any of these studies are. And we asked the question, can we do a better job? So currently, the gold standard is the molecular signature's hallmark KRAS signature. And we said, how good is the signature? Long story short, we generated our own signature. And then when we compared it to the gold standard, which is the hallmark, it was rather troubling that our signature showed very little resemblance to the hallmark signature. At this point, we said, are we brilliant? Did we generate a better signature? Or are we idiots? Did we generate garbage? For the next year, we then said, let us evaluate whether our signature is garbage or gold. And the bottom line, I would not be telling you this if it was garbage. It turns out to be a quite impressive signature. The other thing we showed is that the KRAS and ERK transcriptomes are very similar. So we merged these two gene sets together. And then we asked, how predictive is this gene signature for a response in the patient? So I go back to that ERK inhibitor trial that I told you. And a subset of patients in this trial did respond with a reduction in CA 19-9 levels. If you look at the hallmark signature, you would have concluded this treatment did not hit the target. However, if you use our signature, you can see that there is actually quite a good correlation between our signature and a response in these patients. We took it to the next step, looking at tumor xenograft analyses treated with different RAS inhibitors. And once again, if you use the hallmark signature, you would have concluded the drugs did not block RAS function. You use our signature, significant induction of inhibition of the pathway. And finally, more recently, we, in collaboration with Mirati Therapeutics, they provided patient data from patients being treated with G12C inhibitors, either colorectal or lung cancer patients. And if you look to the left, if you use initially Mirati used the hallmark signature, and they were very puzzled. They said, patients are responding, but the hallmark signature says our drug did not work. Well, we resolved our dilemma by using our signature, and we said, no, your drug actually did a great job of blocking RAS function. So in closing, these are the two issues that we're facing in the field, and we're making significant progress, but there's certainly a long ways to go. And the current situation is that there are now many combinations with these inhibitors under clinical evaluation, including immune checkpoint inhibitors. So I'll just end by acknowledging people in my group who have been involved in these studies. And furthermore, of course, we could not have done this without a great number of outstanding collaborators, both in academia and in industry. So I'll stop there. Thank you.
Our next speaker is Kathy Giorno. She's a assistant professor in the Department of Cell and Development Biology at Vanderbilt University. Thank you.
Thank you. And thank you to the organizers for the opportunity to share our work today. I'm going to be talking about our work on metaplasia and intrinsic mechanisms by which the pancreas suppresses tumorigenesis. So my laboratory at Vanderbilt studies diseases of the pancreas, including pancreatitis and pancreatic cancer. And we do a lot of basic science using genetically engineered mouse models. And we do this because these are, as you know, both very terrible conditions. Pancreatic cancer has an abysmal 5-year survival rate that's now just reached 12%. And both diseases are typically diagnosed after organ damage and very late in disease progression. And so this makes the study of early events in tumorigenesis critical. And one early event that's common to both pancreatitis and pancreatic cancer is metaplasia. Metaplasia is a pathology term for the replacement of one cell type or tissue type for another. And some common examples that you're likely aware of are Barrett's esophagus, where the lining of the esophagus becomes intestine-like in response to chronic injury, or SPEM, which is a gastric metaplasia, which occurs in the stomach in response to injury, and is the reprogramming of chief cells. In the pancreas, we see acinar to ductal metaplasia as an early event in disease. And to a pathologist, this might simply be the replacement of where there should be acinar tissue with ductal cells. But we like to study this in the context of an acinar to ductal reprogramming event. And so what's been shown is that these acinar cells, which are digestive enzyme producing cells, they have a very specific structure and a very specific function. When they become injured, they actually transdifferentiate to ductal cells. And so they turn off the genes that are required to be acinar cells and turn on a completely different program. And these lesions are thought to function in mitigating disease, repairing the organ. And this is thought to be a critical event in an organ that lacks a known stem cell. So definitely in the mice, but likely in humans as well, these lesions will resolve when the injury is lost. And so we don't think of this as an event that's required for pancreatitis. We think of this more as an ongoing repair event that occurs in the pancreas, whereas you're generally using your pancreas, it's exposed to different kinds of injury events, and this is how your pancreas heals. However, in the context of an oncogenic mutation, and we just had a great discussion on oncogenic KRAS, these lesions will form, but they can't resolve. And instead they persist and eventually progress through several grades of neoplasia before eventually forming cancer. And so this takes quite a long period of time. And the more we study the pancreas in terms of pathology, the more that we see that really anyone can have metaplasia and impairment. There have been many studies of accident victims or even organ collection from aged participants lacking pancreatic cancer that have these Pannin lesions and never have cancer. And they're doing really interesting studies at Johns Hopkins now, quantifying the number of Pannin in a given person, and they find there's quite a lot of lesions. And so are these Pannin lesions inert, or are they actively suppressing tumorigenesis? And so here's some pathology examples of the process I described, where these acinar cells become ductal in response to injury or oncogenic mutation. And then they proceed through these increasing grades of Pannin, which are characterized by a loss of polarity and increasing nuclear atypia. And it's thought that around the Pannin 2 or 3 stage, we see additional tumor suppressor mutations, which allow for cancer progression. So recently, we asked the question of what cell types or states result from this process of acinar to ductal metaplasia. If the lesion's thought to function in healing and it's a critical event, will we see new markers or programs that come on in this state? And so to answer this question, we used a genetically engineered mouse model where we could inducibly label adult pancreatic acinar cells with yellow fluorescent protein using tamoxifen. And so we did this in adult mice. We labeled pancreatic acinar cells, and then we induced injury using this chemical called cerulean. And while we don't necessarily use this as a model of human pancreatitis, it's a really good tool to induce metaplasia throughout the pancreas. And so we did this for either 2 or 4 weeks. You can see as in comparison to the H&E of the normal pancreas, we have a good amount of metaplasia form here, and lesions are very obvious by 4 weeks. We then excised the pancreas, generated single cell suspensions, and FACS collected these YFP positive cells and then performed single cell RNA sequencing. And you can see here in this UMAP that we identified quite a few cell types and states, some that have been described before and others that haven't. But basically what I'm showing you is that these Asner cells in response to injury form this ductal population that's mucinous, and then they can form either enteroendocrine cells or tough cells as separate lineages. And I think people here are pretty familiar with endocrine lineages because they're very similar to islet cells. But we found a lot of heterogeneity in this population, including the expression of a number of hormones that are expressed either in islets or in similar cell types in the intestine. We also saw the formation of a population of tough cells, which have been shown to drive Th2 immunity in other organs, and I'll discuss those in more detail later. To visualize this lineage tracing and transdifferentiation event, again, we labeled these pancreatic Asner cells with yellow fluorescent protein. So you can see here that the amylase positive cells are YFP positive, whereas the signal is lacking in this chromogranin A positive islet. But in response to injury, we now see that both Asner cells and these ductal populations, including these chromogranin A positive cells, are retaining the signal. And so what this means is that Asner cells have been traced into these endocrine populations. We find these populations present in human pancreatitis as well. These are samples collected at Vanderbilt University from chronic pancreatitis patients. As compared to nearby normal, we identified solitary hormone-producing cells associated with metaplasia, these pancreatitis patients. And this includes somatostatin, serotonin, ghrelin, and pancreatic polypeptide. We also found a significant population of tough cells, and this is a cell type that people tend to be less familiar with. They are solitary chemosensory cells. They're analogous to taste cells in the way that they express taste receptors. So they express sweet and bitter taste receptors, and they're thought to sample or taste the gut, but they don't aggregate in buds. They're normally found in the respiratory and digestive tracts, but they're a very rare cell population. In the murine intestine, they make up only about 0.4% of the epithelium. They can be identified by their structure, which is shown here in this electron micrograph, by these tall, blunt microvilli and deep actin rootlets. They express both inflammatory and neuronal effectors. And the most well-known function was discovered in 2016, where they were found to participate in the weep and sweep response in the gut. And so this is a response where tough cells induce fluid secretion and mucus secretion from goblet cells to clear parasitic Helminth infection, right? So what are they doing in the pancreas? Again, we performed lineage tracing, where we labeled pancreatic Asner cells with yellow fluorescent protein using the same model. We then induced injury, metaplasia, and tough cell formation with cerulean. And as you can see here, the tough cells are labeled with phalloidin, which marks the actin rootlets and the microvilli. And we found that 99% of these tough cells are actually derived from Asner cells. So this is a pretty substantial reprogramming event in the pancreas. Tough cells are also present in human chronic pancreatitis. And so on the left, this is a pretty impressive study that came out of Germany, where this group did single nucleus RNA sequencing on human pancreata. In the UMAP on the left, they collected 300,000 nuclei from normal pancreata, and they generated an app that you can use online to look for your favorite cell types and your favorite markers. But you can see here, they didn't identify any tough cells, but they did in this much smaller sample set taken from pancreatitis patients, which only constituted about 2,000 nuclei. But even within that small data set, they were able to find a population of tough cells confirmed by the expression of known markers. And ourselves and others have also done immunofluorescence to confirm the formation of this population. So as compared to the nearby normal pancreas, these metaplastic ducts express here this marker is phosphogrenin, solitary tough cells in the metaplastic lesions. So given that we've identified tough cells and enteroendocrine cells as substantial populations in injury-induced metaplasia, we next asked if KRAS-induced metaplasia also contain these populations. And so what we did was align our single-cell RNA sequencing data set to one generated by another group using mutant KRAS. And so what this group did was a very similar experiment where they labeled adult pancreatic Asner cells with tomato, which is a red fluorescent protein. at the same time activating mutant KRAS. They then allowed the mice to age for, I think, about 9 different time points, some including cancer. They collected the tomato-positive cells and performed single-cell RNA sequencing. And at a quick glance, you can see that we have a lot of the same populations that they found in the KRAS model and a number of different populations as well, which we talk about in this publication. But you can see here that KRAS also induces tough cell and enteroendocrine cell formation. And so we performed a number of studies to quantify these populations in metaplasia and neoplasia in mouse models in human. And as you can see here, synaptophysin, this is a broad marker for enteroendocrine cells and other endocrine populations. But we quantified these cells in metaplastic lesions throughout neoplastic progression. And we saw that these cells make up almost 10% of the epithelium in these metaplastic lesions. They decrease as these neoplastic lesions progress, which is consistent with the loss of differentiation in these models. And we found this both in a KRAS model and in one that also included a mutant P53 allele so that we could look at more aggressive lesions. We also looked at the additional hormones that I described in previous slides and found the same pattern for each hormone. These enteroendocrine cells are also associated with human pannin, and this was shown in this paper as well, where we looked for, in patient samples collected at Vanderbilt University, we looked for solitary hormone expressing cells in these neoplastic lesions, and we were able to identify all 4 hormones that I described previously. And we did some quantification where we looked at, for example, 23 metaplastic lesions and found that a quarter of them express ghrelin and so on. And so we were able to identify all of these hormones as expressed, most of them decreasing once we got to cancer progression, except for pancreatic polypeptide, which we find really interesting but have no explanation for at this point. Tough cells are also found in murine pannin. So we're seeing tough cells and enteroendocrine cells in metaplasia associated with injury and KRAS. These images are not as pretty, but they were collected in 2014. But what we found here using this marker DCLK1 is that tough cells formed in response to KRAS and were most abundant in metaplasia, similar to enteroendocrine cells, and then decreased in abundance with disease progression. And so collectively, this data suggests that up to a quarter of the epithelial cells that form in metaplasia are either tough cells or enteroendocrine cells. So to summarize the first half of this talk, what I've told you is that metaplasia is an early event in both pancreatic injury and oncogenic mutation. It's the transdifferentiation of these very plastic acinar cells into both tuft and enteroendocrine cell populations. And so what we're seeing is the change of a very differentiated specific function acinar cell to other cell types that have very different functions. These enteroendocrine cells are heterogeneous and they express several hormones. And this could have very interesting implications for crosstalk between these lesions and islet cells in pancreas diseases. But the big question here is whether or not these populations are functional and if they play a role in disease progression. And so we have a number of projects in our laboratory looking at these enteroendocrine cells, but what we've mostly studied is the function of these tough cells in disease progression. So to better understand their role in tumorigenesis, we took advantage of the fact that these cells are known to be reliant on transcription factor PAU2F3 for their formation. So we generated a mouse model where we simultaneously ablated PAU2F3 specifically from the pancreatic epithelium while activating mutant KRAS. And you can see in this H&E that this did not inhibit tumorigenesis. We still had neoplastic lesions. We see a loss of transcription factor PAU2F3 and a loss of epithelial COX-1. This is typically expressed in immune cells, but in the epithelium, it's very tough cell specific. And we were surprised to find that this actually accelerated tumorigenesis. And this is contrary to what we and other groups thought would happen. Some people thought this cell type represented more of a stem cell, while others thought that the expression of these COX proteins meant that they would drive tumorigenesis. So we were very surprised to find that we actually accelerated tumorigenesis when we ablated this cell type. So you can see here when we aged the mice, by 6 months, we had significantly more Pannin 1B in these are the tough cell knockout mice as compared to KRAS. We had Pannin 2. And then by a year, we had significantly higher incidence of cancer. To better understand how tough cells are inhibiting tumorigenesis, we performed another assay. where we took either KRAS mice or KRAS mice without tough cells at about 6 weeks of age. And this is before they formed any lesions. We gave them a short course of cerulean to induce injury, and then we waited only 2 weeks. And you can see that by 2 weeks, we had significantly more injury. We had significantly less normal tissue by amylase staining, significantly more extracellular matrix deposition by hyaluronan. And within even this just really short time frame, we were able to see accelerated pannin development, where we had significantly more pannin 1B in the tough cell knockout mice and higher incidence of pannin 2. And this is only a couple weeks after injury. Due to their known role in influencing immune cell infiltration, we use single cell RNA sequencing to better understand the microenvironment. And so what we did was collect the stroma from either KC or KPOC mice using FACS collection and perform single cell RNA sequencing. And by doing this, we're able to directly compare fibroblasts in different immune cell populations. And I'm going to show 2 here. As compared to the KRAS mice, the KPOC mice or tough cell knockout mice with injury had a significantly larger MyCAF population, which is either a fibroblast type or a cell state, depending on what camp you belong to. But they express alpha SMA, and one major function is to deposit extracellular matrix. So we saw significantly higher abundance of these alpha SMA or ACTA2 positive fibroblasts. When we assayed the macrophages, we found significantly higher expression in our knockout mice of all of these different cytokines, which suggests that these macrophages are more pro-inflammatory. So collectively, between these 2 cell types, and these data are now published, we found that the stroma is more pro-inflammatory in the tough cell knockouts. So do they play a role in inhibiting inflammation and therefore inhibiting tumorigenesis? So to get a better idea of how tough cells might function in this context, we created transcriptomes of these cells. We took advantage of the fact that they express cyclic F, which is typically an eosinophil marker on the cell surface. And we FACS collected cyclic F EpCAM positive cells from the pancreata. Again, they're rare, so we were only able to get 1 to 200 cells per pancreas. So we use small cell number RNA sequencing approaches, in this case, SMART-seq2, to generate deep RNA-seq profiles of these cell types. And we were able to do this successfully. We used these profiles and did network analysis to identify pathways that are specifically upregulated in this cell type. And consistent with what has been published, we found a lot of immune cell activation pathways, and then eicosanoid biosynthesis and arachidonic acid metabolism. And so as I showed you before, these COX proteins are actually pretty reliable markers for tough cells. So eicosanoids are lipid mediators that are known to influence inflammation. And as a whole, they're generally considered to be pro-inflammatory and pro-tumorigenic, though it's a really large, complicated family of lipids. And so in the context of pancreatic cancers and other cancers, synthases like COX-2 and species such as prostaglandin E2 are known to drive tumorigenesis. But until this point, it was unknown what eicosanoids would be present in pannin-bearing pancreata. And so we took whole tissue and submitted it to UCSD to do eicosanoid profiling. And we're actually surprised to see that the most differentially upregulated prostaglandin was prostaglandin D2, which is relatively understudied, and there's only a few papers that describe it in the context of cancer, but they generally describe it as inhibiting tumorigenesis. And so PGD2 forms downstream of these COX proteins and then this synthase HPGDS. So we assessed both our KC pancreata and age-matched tough cell knockout KRAS pancreata, and we found significantly lower levels of prostaglandin D2, suggesting that a large amount of this eicosanoid is generated by tough cells. Other cell types that can make this are immune cells and include macrophages, which could make up for the rest of the PGD2 that we see here. To determine if epithelial or tough cell specific PGD2 could play a role in tumorigenesis, we created another mouse model where we floxed this synthase, which is the HPGDS allele. And these pathways become kind of like an alphabet soup where you're just saying letter after letter. But the HPGDS was knocked out specifically in the pancreas in the context of activating KRAS to ablate epithelial PGD2. And you can see here by this RNA fish that we now lack expression of this synthase in the epithelium. And so we perform mass spectrometry with UCSD. And as you can see here, as compared to the KRAS mice, these are KRAS mice lacking epithelial HPGDS, and this is in the context of cerulean injury. We see significantly less PGD2 in the pancreas. And then very similar to our tough cell knockout mice with the same protocol, we see within this 2-week time frame, significantly less pannin 1A, significantly more pannin 1B, and even some pannin 2. So epithelial loss of PGD2 is sufficient to accelerate tumor genesis as well. You can see here in this immunostaining that tough cells still form. In the HPGDS knockout, we still have POW2F3, we still have COX-1 expression. But just like the tough cell knockout, we have significantly more injury. We have less amylase staining and more extracellular matrix deposition, in this case by trichrome, which marks collagen.
Just so you have 1 minute left.
Sure. So to understand better what the role of PGD2 might be in activating the stroma, in this case, we collected stellate cells from the pancreas, activated them in vitro, and then treated them with PGD2. And this was sufficient to decrease alpha SMA expression, which the gene name is ACTA2, and these collagen synthase enzymes. We did a similar assay with macrophages where we activated bone marrow-derived macrophages in vitro with LPS. And then when we treated them with PGD2, we decreased this interferon signaling pathway. So in both cases, it reduced pro-inflammatory gene expression. This slide just shows that both HPGDS and tough cells are present in human pannin. And then this is summary 2. And so what I've shown you is that in response to oncogenic mutation, acinar cells transdifferentiate into metaplasia and these neoplastic lesions, and that these are very heterogeneous. They contain tough cells and enteroendocrine cells. And these lesions still progress, but it's very slow. So only about 10% of these KRAS mice have cancer by a year of age. And so the idea that I'm posing here today is that a number of these cell populations that are differentiated that form from mutant KRAS are actively inhibiting disease progression. And what I've shown you is that when we ablate tough cells, and I think it'll be the same for certain endocrine populations as well, is that we get a more pro-inflammatory microenvironment and that this causes rapid disease progression to cancer. And so in the future, the way that I like to think of a lot of these knockout mouse experiments is, are we knocking out a pathway that's common to all of the neoplastic cells, or are you specifically affecting the physiological function of one of the cell types that forms in these lesions? And with that, I'll finish. I'd like to thank my laboratory and Vanderbilt University collaborators for helping me put out the single cell RNA sequencing story that I showed in the first half. And then my colleagues at the Salk Institute from when I was a postdoc in the Wall Laboratory. And we have quite a bit of funding and open postdoc positions available. Thank you.
We'll open it up to a few questions from the audience.
This is a fantastic session, and I have slightly less than 1,000 questions. Just an interesting comment is that there are, in the human pancreas, pancreatic polypeptide is more in the dorsal pancreas, whereas glucagon is in the ventral pancreas. And I'm wondering if there's a difference in biology related to pancreatic polypeptide, which could be looked at in humans based on where the tumor forms and how it develops. So that's one of the questions, that whether or not you look at where the tumor came from and what endocrine cell type is normally expressed in those areas.
That's a really.
Oh, yeah, I have to turn it on.
I figured out the microphone. That's a really interesting comment, and I actually didn't know that until yesterday. So in the future, we will make sure to separate the two and see if the abundance changes. For speaking to the islet biologist at Vanderbilt, pancreatic polypeptide is at least considered to be a very minor population in the islets. And my assumption was regardless of location, but I'll definitely rethink that now. And even the islet biologists seem to have kind of not put very much focus on this population. And there are groups in Europe that are picking it up now. And they're finding that, I think in response to injury, there's more bihormonal populations where delta cells are also now expressing pancreatic polypeptide. And there's some suggestion that it's a response to injury and that it might also be anti-tumorigenic.
Jens Kort from AbbVie, fantastic talk. Just wonder whether you're also going to look at, T regulatory cells and other T cells in your model. We looked at the macrophages here. We know macrophages are a big aspect we can see in development of cancer and inflammation. But I wonder, is there another story to obviously the other arm of the immune system?
That's a fantastic question. And so we did look at the T cells in that particular model. And we didn't see a lot of major changes. I think one, because we didn't capture enough, and two, because they might be mostly influenced by KRAS. But we have a number of injury models set up. And so what we've found there is that tough cell ablation actually causes larger amounts of injury, and that seems to have an effect on T cells. And so what we're doing is looking for a role for Th2 immunity. The tough cells in the injury environment express cytokine IL-25, but they don't in the KRAS setting, which was a pretty interesting difference. And the KRAS tough cells were one of the first that don't express IL-25. So now what we're doing is seeing if tough cell ablation in the context of injury and then the loss of IL-25 impacts eosinophil infiltration, mast cell infiltration, and if it disrupts this Th2 immunity that might be required to kind of quell inflammation in the chronic setting.
I failed the IQ test early in the morning in terms of turning on a mic, but enjoyed both talks. I had a question actually for Dr. Deer. KRAS, at least from those of us who are in the early detection field, is always with our Panin model an early mutation. Do you ever think, I was concerned about all the toxicity you're describing in these trials, not sure if some of that's related to the classic chemotherapy plus the inhibitor or in the combination. Do you ever think there will be a role for a preventive agent since KRAS is early on in the Panin thing, or do you think toxicity is going to prevent that?
So yes, toxicity, normal tissue toxicity is being seen with the mutation selective inhibitors, which is not exactly expected. We anticipated that because these are mutation specific inhibitors and the mutations only occur in the tumors, these could be the perfect magic bullet for cancer cells. But there is normal tissue toxicity, and we don't know the basis of it. So I think one of the issues that we face moving forward is to understand why there is normal tissue toxicity. And then if we can eradicate it, and if we can truly make these mutations selective, then yes, potentially these might have a preventative role because patients should be able to take these drugs and it should only impact the tumor cells and not have any impact on any normal tissue. That's obviously easier said than done, but that's one of the challenges that are faced right now. And we don't understand where the toxicity is coming from.
Any other questions? One up here in the front. And then Zobeda after that. This gentleman first and then Zobeda. Sorry.
Hi, good morning. Great talks, both of you. I have a question for you, Dr. Dare. in terms of KRAS targeting drugs, what are your thoughts of the comparative efficacy? And it may be too early to tell of the different mechanisms, whether it's degraders versus inhibitors or immune modulated.
I'm sorry, could you just repeat that?
The comparative efficacy between the different methods by which KRAS G12D is being targeted now, degraders versus inhibitors or versus immune.
Okay, so if I think I caught your question, you're asking about a degrader versus an inhibitor. Yes.
What's the difference in efficacy?
What's that?
What's the difference in efficacy between them?
Yeah, so that's a great question. Many people would like to think a degrader should be a superior inhibitor, in particular because some of the resistance mechanisms involve second site mutations. So the degraders may take care of those. Of the G12D inhibitors that are either preclinical or clinical evaluation, one is a degrader, one is a non-covalent inhibitor, and one is a covalent inhibitor. And then we have a pan-RAS inhibitor. So we have 4 chemically distinct inhibitors, all targeting G12D. We can speculate as scientists which ones might be the best. But I would say, having been in this business long enough, we typically are wrong when we speculate. We're just not smart enough. At the end of the day, the patient will tell us what the answer is. And I say that because the pan inhibitor, which the experts predicted would be a disaster, that patients would croak. That seems to be working the best, and nobody handicapped that. So I think as scientists, we are humbled by the fact that we are still pretty ignorant.
Hi, I was curious, you said it took a long time to find out that unexpected pocket to be able to target. Why was that?
So why did we not find that pocket before, despite the fact that hundreds of crystal structures of RAS have been published? One has to appreciate that crystallography is a snapshot of a dynamic molecule. And I guess our cameras are not good enough. It did not capture that pocket because RAS is actually quite dynamic and it goes through many, many different phases. The only reason that pocket was eventually identified by Kayvon Shokat is because his molecule bound in that pocket and stabilized it so we can finally see it. But otherwise, it was a very transient state that was simply not captured despite the fact of publication of hundreds of crystal structures.
I think that goes to show a lot of us that we don't necessarily have to believe, or it's okay to think outside the box and go against dogma, which is one of the things that one of my mentors always told me. And I'm saying this because there's a lot of trainees in the room.
Any other questions? Yes, one.
Oh, great. Great presentations, fascinating. One question I had for you is when you were measuring the specific cell, sorry, I forgot the name, the one that's rare, the tough cells, thank you. When you were doing the single cell RNA-seq, was there a difference in an expression level to show the possible degradation or termination of those cells? Was there something that could be used to detect when they are going to evolve to the next pan-end?
When they're going to what?
Move to the next. So basically, because when they die off, as you said, it increases disease. Is there something in the expression profiles that you could see that is indicative of that cell death?
No, that's a really good question. Frankly, we were lucky to be able to get healthy live cells out of the pancreas to begin with. That is a great question. We have a number of theories of what's going on that we haven't tested yet. And I do get this question a lot. It's a great question. It's possible that the population that makes tough cells is kind of similar to the intestinal stem cells, right? It's receiving different signals and then it pops out an endocrine cell or a tough cell. It could be that those signals are changing We see less tough cells when we have a P53 mutation or a SMAD4 mutation. So that could be related to the differentiation of the tissue. They could be dying off at the expense of other populations, which is what we do see in KRAS. We see an expansion of the mucus producing cells and the formation of pit cells and the loss of these cells. So we don't know, and we don't see anything different. And we looked at tough cells from 10-month-old versus 5-month-old, 3-month-old mice, and we didn't see any major differences in the gene signature.
Does the KRAS the molecular system be converted or no?
To form or to go away? Both, really. So we've seen tough cells form in response to cerulean, in response to KRAS, in response to duct ligation in the aster tissue, in response to chronic salmonella infection. So yeah, so it seems like really any kind of injury that persists in the pancreas for several weeks, you'll start to see tough cell formation. So we don't see it in acute protocols. We don't see it after only a week or so. You have to kind of have a much more mature metaplasia, and then you get tough cells, enteroendocrine cells. And it's consistent with all these different models. And then we're quantifying it now, but what we've seen is that when acinar cells start to undergo metaplasia, they're very pro-inflammatory, and we see them directly associated with a lot of immune cell types. But when we get these really stable lesions, we actually see less immune cell infiltration. Yeah, so we think it's still there, but we think that it's inducing homeostasis, and that's what allows these lesions to persist. But clearly they change over time, and you start losing cells, and then you get progression.
Okay, one final question.
Just about tuft cells and their geography and the development of them within the injury. Is there a timeline, sort of like you can see them more in the head of the pancreas moving to the tail over time, or are you able to capture that? Are they everywhere at the same time? That would be just.
No, that's a great question. We haven't separated it by head or tail. We generally see more when we see more injury. So the assumption would be that there's more in the head than the tail, because other groups have shown that there's more lesions that form in the head in response to the cerulean and the neoplasia, but we haven't separated them.
Well, thank you for a great discussion, and we'll move into the second session where we'll invite the other moderators up.
Okay, can you hear me? Okay. We're going to switch gears to the clinical part. This was a great session with a basic science from a clinician perspective. So it's my pleasure to introduce Dr. Amy Lucas. She's a professor of medicine of the Division of Gastroenterology at Mount Sinai. Amy, the stage is yours.
Thank you so much. Thank you to the organizers for inviting me to speak today on the status for defining risk groups for pancreatic cancer beyond hereditary etiologies. These are my disclosures. I do receive research and/or consulting support from these groups. The objectives for this morning will be to identify risk factors for pancreatic ductal adenocarcinoma, to describe novel methods for assessment of pancreatic cancer risk beyond monoallelic germline variants, and then think about the integration of pancreatic cancer risk as well as early detection. So I think it's important to take a step back here and really think about the difference between risk prediction versus early detection. So when I think about risk prediction, I really think about the modifiable and unmodifiable risk factors. And we know about these modifiable risk factors: tobacco exposure, heavy alcohol use, obesity, etc. And unmodifiable risk factors include age, sex, race, family history, as well as genetics. And then when we shift to early detection, we can start to think about imaging for early detection, biomarkers in a variety of spheres. Diabetes can sometimes be thought of in this way, as well as clinical data and EHR data. So when we think about our general population, it's impossible as of now to screen for pancreatic cancer in the general population. Even if we had a perfect test with basically near perfect sensitivity and specificity, the number of false positives that would generate and the downstream costs would make this approach unfeasible. But the key is to really start to identify that more narrow population that do have increased risk factors so that we can get to and intercept that 1.6% of individuals who will go on to develop pancreatic cancer over their lifetime. So what are these risk factors? We've known about them for years. Here, I just show you some data on obesity and alcohol. We've seen this in single center studies and observational studies, case control, meta-analyses, et cetera. But what you can see in these studies is that the odds ratios for development of pancreatic cancer are relatively modest, oftentimes in the 1.2 to 2 odds ratio range, and therefore insufficient for detecting risk and predicting risk of pancreatic cancer. We also have diabetes, which we know is a risk factor for developing pancreatic cancer. But we've also seen several papers that it's also an opportunity to predict pancreatic cancer at various intervals prior to the development. And on the top, we see data from Suresh Chari's group that I think we've all seen, where glucose levels can be elevated up to 3 years prior to diagnosis, with frank diabetes happening 6 to 12 months prior to diagnosis. And then on the bottom, data from our group showing that in a diverse population, hemoglobin A1C as well as glucose starts to be elevated 3 years prior to diagnosis. And the patients start more obese, but about 2 years prior to diagnosis, start to have more pronounced weight loss. There are also interesting associations between age and risk factors for developing pancreatic cancer. So this study actually looked at patients with 5 known risk factors for pancreatic cancer. And what you can see is with acquiring more risk factors for pancreatic cancer, you can see increased risk for pancreatic cancer development. But when you look at a younger cohort, so less than 60 years of age, those risk factors are much more pronounced. And that is more attenuated in older individuals over the age of 70. This group also looked at the age-specific proportion of pancreatic cancers that were attributable to these risk factors. So these are the pancreatic cancers that never would have happened had those risk factors not existed. And what you can see is that for younger individuals, generally speaking, that population attributable fraction is higher than it is for older individuals. We also see interesting trends in time and sex-specific trends for the development of pancreatic cancer across different age groups. So we all know that pancreatic cancer is increasing in the United States. And what we can see in all ages is that the average annual percentage change, so this is sort of the annual increase in pancreatic cancer, generally is slightly higher for men than for women. But if you stratify by age, over or under, let's say 55 in this instance, you can see that the trend holds for men over the age of 55, but it's perhaps more pronounced. But if you look at younger individuals, you can see that women actually have a higher average annual percentage change than men. And so this begs the question of whether or not there are sex-based disproportionate exposures to either known or unknown risk factors. And perhaps we can think about birth cohort effects or gender-specific impacts. We also know that pancreatic cancer is influenced by the number of affected first-degree relatives. And although I'm supposed to stay away from the hereditary setting, we also know that a number of hereditary syndromes predispose to pancreatic as well as other cancers. And efforts have been made in this hereditary setting to assess risk for pancreatic cancer. But to date, these have really been insufficient. But what about individuals without a strong family history? And so moving away from those monoallelic germline variants. A number of studies have now been published, GWAS studies, identifying a variety of different susceptibility loci for the development of pancreatic cancer. And the benefit of these GWAS studies is that they can be combined to generate polygenic risk. However, the trouble with polygenic risk scores, at least thus far in pancreatic cancer development, is that they've really been insufficient to predict the risk of pancreatic cancer. An additional concern is that many of the GWAS studies are performed in predominantly European ancestry populations. And so then there's also questions about how those might apply in more diverse and multi-ethnic populations. I'll take a moment to focus on this study in the top. This study came out of Europe and looked at pancreatic cancer patients, about 3,600 pancreatic cancer patients and 5,700 controls in the Pandora Consortium. They confirmed previous SNPs for risk of pancreatic cancer, ABO blood group data, but they also had the availability of exposure data and specifically tobacco and diabetes. And they further performed a validation step using genotype data from other previously published cohorts. Once again, the PRS, when we compare the highest to lowest quintiles, performed rather modestly with odds ratios in the 2.6 to 2.7 range, and area under the curve in the 0.6 range. However, they also were able to integrate these clinical data, such as tobacco and alcohol. And you can see that in both the unweighted and weighted scores, integrating tobacco and diabetes, improved the risk stratification. And the authors argued that this magnitude of increased risk was really actually quite similar to that we see for monoallelic factors such as BRCA2 or ATM, for whom surveillance protocols might be recommended. The group that also looked at the age-dependent risk factors for pancreatic cancer also generated a PRS score based on GWAS data. And interestingly, they found that the PRS performed differently in different age groups. So it performed best at age less than 60. This other group evaluated pancreatic cancer risk in the UK Biobank. UK Biobank is a large data set, again, in the UK, so predominantly European ancestry. And they had available 1,000 cases and about 10,000 controls. They were able to also evaluate diabetes subgroups, so those without or with diabetes, longstanding diabetes, or new onset diabetes. And again, they saw relatively modest odds ratios with these risk factors for the development of pancreatic cancer. They generated 5 PRS models that included SNPs from previous studies, including the one that we just looked at, and then also created a combined PRS, which performed best, but again, with an area and a curve around 0.6. Because they had the diabetes data, they were actually able to stratify their PRS by individuals without diabetes and compared to those with diabetes. And they actually saw that the PRS performed much better in individuals with diabetes. However, sensitivity for the PRS remained low, specificity was a bit higher, but if you look at the positive predictive value, particularly with those with diabetes and really new onset diabetes, the positive predictive value was impressive. Sorry, I'm having clicker problems. So what have other groups done beyond polygenic risk scores? This is an interesting paper that was published just recently where they evaluated patients in the Danish National Patient Registry, so 6 million patients, which included 24,000 pancreatic cancer cases, as well as 3 million US veterans, which is 3,900 pancreatic cancer patients, and applied AI methods to clinical data to be able to predict risk of pancreatic cancer. It's jumpy. So what they did was they essentially took their patient population, extracted those clinical diagnoses, and time-stamped them over various different periods. They had a testing set, a training set, and development set. And they were able to look at clinical disease trajectories over time to development of pancreatic cancer, to death or no development of pancreatic cancer, apply machine learning techniques, and then be able to predict pancreatic cancer development. These are the risk factors that pulled out when thinking about their algorithm. So these are the known risk factors in the Danish cohort as well as the USVA cohort. But I'll draw your attention here to the left when we look at those risk factors, because some of them we would think about as true risk factors for pancreatic cancer. So obesity, probably diabetes, et cetera. However, some of these purported risk factors might actually just be a signal that a pancreatic cancer is there, but just not yet detected. So acute pancreatitis or weight loss might in fact just be a signal that there's a pancreatic cancer waiting to be detected. When applying their algorithm, they were able to look at a variety of different time points prior to diagnosis. So they were able to look at 3 months, 6 months, 12 months, 36 months, and 60 months, and be able to look at the performance of their algorithm. And they actually found that it performed pretty well, it performed better, closer to the time of diagnosis, and perhaps a little bit less well up to 3 or 5 years prior to diagnosis. But still, this is rather impressive data. And so essentially what their algorithm and description is that they can take a number of patients for assessment, and we have these data available in our electronic health records, apply their error minimized neural network, follow patients over time, and evaluate their risk over time. And once an individual exceeds a certain risk threshold, you can categorize them into a high risk cohort compared to a low risk cohort, and then apply cancer surveillance strategies. And therefore, you've defined a new group of individuals who are at increased risk of pancreatic cancer. So what are other ways that we can leverage data that we already have to be able to detect pancreatic cancer earlier? Well, we have a lot of imaging data that are available in our EHRs and on our systems. So this paper actually looked at a number of individuals who had pre-diagnostic CTs. So these were CTs done somewhere between 3 months and 3 years prior to a diagnosis of pancreatic cancer, and they were done for another reason. They compared them to control cohorts, extracted radiomics features, and then applied machine learning, and essentially were able to predict which individuals had pancreatic cancer compared to those who didn't. And then, of course, we have our biomarkers for early detection. We have thousands of papers that have been published over the course of the last decade. People are looking at urine, looking at stool, looking at microbiome. And then there are a number of blood-based approaches. I include some of them here for early detection. And then, of course, also pancreatic juice for early detection. So what is the current status for defining these risk groups? How do we get from that general population down to that smaller population with risk factors, ultimately to identify and intercept that 1.6% who have lifetime risk? Well, we can think of this as a strategy where we identify a baseline risk, and that could either be genetics or monoallelic or polygenic risks. We can integrate our modifiable as well as unmodifiable risk factors to be able to look at individuals at particular points of time, apply early detection, whether it's EHR data, clinical data, radiomics data, et cetera, and then move on to prevention as well as confirmatory tests or surveillance in that population. So to conclude, currently our known risk factors are really insufficient to predict pancreatic cancer. The impact of risk factors, interestingly, might change with age. We might see effect of a birth cohort effect. And PRS, as it stands now, seems to be currently insufficient alone for detection of pancreatic cancer, although the combination of PRS with risk factors does improve risk stratification. Just a word of caution when we're looking at this in the literature, some risk stratification may in fact be early detection, but perhaps that's neither here nor there, as a combination of PRS, risk factors, early detection does seem to have great potential. And one could envision a future that might include a combination of precision medicine approaches, risk factor identification and modulation, and early detection and prevention. I thank you for your time and looking forward to the panel discussion.
Better now? Okay. Thank you, Dr. Lucas. This is a wonderful talk. Now I'd like to introduce Dr. Panicia from right here in Pittsburgh.
Good morning, and thank you. First of all, I would like to start and thanking the organizing committee for the privilege of the podium. I have no conflict of interest related to this talk, and I've been tasked with the clinical prediction to response to neoadjuvant chemotherapy. This is truly a field in evolution, but it's a very familiar curve for pancreatic cancer survival. When you look at this curve, you see two very distinct populations. There is a 30% of cohort that survives long-term, at least beyond 5 years. And these are usually patients that have good response to chemotherapy, excellent surgery, and so forth. But what is very concerning is the early part, the left side of the curve, this early tumor recurrence, commonly defined between 6 months to 12 months after optimal oncologic outcome. These are patients that receive chemotherapy, have surgery, but still have a death of early recurrence within the first 6 to 12 months. And you will argue this is really not ideal from a surgeon standpoint. You put this patient to maximal therapy and they have such early recurrence. We cannot, we need to wonder what happened here. Is that a late diagnosis, is a different tumor biology that we gave the wrong therapy? And it's pretty clear now that the field of surgical oncology, especially for surgery for pancreatic cancer, is moving beyond just anatomy definition, and we need to account for biology. This is quite an important paper from 2017, really redefined the concept of resectability, including a biological characteristics, which is mostly characterized by elevated CA19-9, among a few other factors. Not to dwell in the merits of the neoadjuvant chemotherapy, which is not the goal of this talk, but this is a good example of a validation of the new criteria. What happens if you take patients directly to surgery without accounting biology? These are about a cohort of 400 patients divided between resectable, borderline resectable by anatomic criteria. And you see a resectable tumor without any biological component, as it is currently measured, as a reasonable survival and early recurrence rate at 6 months of only 16%, which is quite good for pancreatic cancer. But what happens if you move beyond? You take borderline resectable by anatomy, 25% at 6 months, and then when you include the biological conditional factor, you start having recurrence at 6 months of 30 to 60%, which is quite catastrophic, I would say. These are a Kaplan-Meier curve that really represent this concept graphically. You can see how that inclusion of the new criteria really separates the curve in 4 really distinct cohorts. But what is interesting on the right side, you have the green and the red curve, which is a borderline resectable by anatomy and a borderline resectable only based on biology, such as elevated CA19-9 above 500. You see how those two curves overlap. It really is a testament to the importance of those measures. And when you combine both borderline anatomy and biology, you have a median survival of less than 10 months. Here in Pittsburgh, we have done about a decade work on CA19-9, and I'm just going to show you the summary of all of this work. We took our cohort of about 6 or 7 years, all resectable, borderline resectable patients. They all received neoadjuvant chemotherapy. We excluded the patients that were CA19-9 non-secretors, so this is a pretty uniform cohort in terms of secretion of CA19-9. And what we look at was survival and effectiveness of chemotherapy based on not on cycle, not on months on therapy, but a CA19-9 response. And by our analysis, what we define as an optimal response threshold is a decrease of CA19-9 by greater than 50% and a normalization. But what is important here that we only saw a survival advantage, now 14 months versus 20 to 27, only in the optimal response. Receiving therapy without evaluation of efficacy is really does nothing for this patient. It's probably because all the trial that have been done, why all the trial that have been done really did not show a significant advantage on neoadjuvant chemotherapy, because none of those trials measure effectiveness. But what is the problem with CA19-9? It's a good marker, but it's not a perfect marker. And about 10 to 20% of the population really does not secrete CA19-9. So it leaves you blind in about a fourth to a fifth of our entire cohort. This is an interesting study that was recently published using a precursor of CA19-9, which is called UPAN2. This is nothing new, has been around for a decade or two, but finally we were able to package this in a useful format. You can see what the study did, took all patients, secretor and non-secretor, defined the secretion stage by less than 2 units per milliliter, which is truly the non-secretor population. Then you have the low secretor, which is a different category between 2 and 37. And then you have the 37 to 500 and 500 above. The first question that they answer is there a biological difference between a secretor and a non-secretor? And the answer is on the left side. They have identical survival when you adjust for other factors. So a non-secretor is not a different cancer compared to a secretor. But when you go on and break it down by CA19-9 level, you see the similar curve to what I showed you before. And when you find the optimal threshold for DUPAN-2, which in this paper was defined as above 2,000, a value above 2,000 unit per milliliter, correspond to a value of above 500 unit per milliliter of CA19-9, really bringing you back to the international consensus. So this is now an option to judge response to chemotherapy and effectiveness potentially in non-secretors. What about imaging? Imaging is important in pancreatic cancer, but mostly for diagnosis and for surgical planning. It's well known that CT scan complementary tomography by RECIST criteria really does not help us decide who's a candidate for surgery and who's responding to chemotherapy unless you see obvious progression. This paper was just published maybe 2 months ago, experience from the Mayo Clinic. And I chose this paper in particular because these patients are the population that we care about. This is the locally advanced borderline resectable tumor, heavily pretreated with chemotherapy, maximal therapy, FOLFIRINOX or Gemabraxane, which is a modern regimen. 90% receive chemotherapy. So these are patients that likely were not resectable to begin with. Now here you'd really need a response. And they found that PET scan is actually a very good marker for response in this situation. In their cohort, only 3% of patients had a PET silent tumor. They defined the response criteria based on background liver and pancreas, and they found an accuracy of response to chemotherapy up to 84%. But what's striking of this paper is the following. When they finally performed surgery, they found a major pathologic response occurring in 75% of patients that had major PET response. And this was completely independent by CA19-9 response. So in this cohort population, heavily pretreated, when you have a PET signal, PET scan could be very helpful to help us decide who needs to undergo surgery, who's responding to chemotherapy. We obviously know pancreatic cancer is not a unified disease. This is now all data. We have, if you look at a multi-array analysis of pancreatic cancer cell, you see 2 clusters of gene, a basal type, a classical type. A long shot is stop short, a basal-like type cancer performs worse. Higher mortality, less response to FOLFIRINOX therapy. It's complex to do multi-array analysis. It's expensive. It takes time. This now can be same information can be obtained with surrogates. We can use GATA4, GATA6. This can be done by IHC. This also just published a few months ago in GAT. You can see by simple characterization of GATA4 and GATA6, you can differentiate it for sub cohort of pancreatic cancer. Again, a different survival, very well stratified. But what's important here, that you are able to know who is going to respond to what therapy ahead of time. Currently, we assign FOLFIRINOX or Gemabraxane randomly, mostly based on performance status. But if we had this information a priori, we will most likely avoid FOLFIRINOX in patients that have GATA4 and GATA6 lost. This is a nice waterfall plot. If you look on the panel C on the left side, the light blue color is basically GATA4 and GATA6 loss. And you can see with FOLFIRINOX, there is actually a progression of tumor. Tumor continues to grow. The effect is not true for Gemabraxane. So in this situation, Gemabraxane should be the favorite choice. At least that's what we think now. We're waiting larger study to explain that to us in more clear and fundamental terms. What about liquid biopsy? Liquid biopsy is a very large field. It is evolving, and we've seen in the previous talk, it can be used for screening from diagnosis. We are interested as surgeon in therapy selection and monitoring. Liquid biopsy is composed of different various components. You can have circulating tumor cell, circulating DNA, and extracellular vesicle are the major players. They all different. They all look at different aspects of the tumor cell journey. What is lacking now in liquid biopsy, among other things, is the cooperation of all this information. There are really no studies that account for all 3 aspects of the liquid biopsy in a single patient. Obviously, there are advantages and disadvantages for each, but the story is mostly the same for all of these liquid biopsy. If you do 5 circulating tumor cells, ctDNA or exosome DNA at diagnosis or right after surgery in the first month, your survival is much worse. It brings you back to that 30% recurrence in the first 6 months to a year. And this is constant in every single study that you look at. This is a small study led by Chris Wolfgang when he was still at Hopkins. But the importance of this study, although it's small and is derived from the original cluster study, as they hear, it really tries to differentiate the importance of the circulating tumor cell. We know that circulating tumor cells are all the same. There are epithelial type and transitional mesenchymal type. What is important from the transitional mesenchymal type, they are very aggressive. They signal a phase of the tumor that is able now to metastasize, to prime different organ for metastasis. In the study, they try to answer the question, can we affect management? Usually the comment anytime you bring up liquid biopsy for therapy is like, what do we do with the information? Right? Yes, we have to then consult patient. It becomes more of a burden than a utility. And this is in a small little capacity, answer a bit of that question. Obviously, show that if you had transitional mesenchymal type CTC right after surgery, your survival is much worse. But the question is, can you mitigate this? And we know the adjuvant chemotherapy helps pancreatic cancer, but from the ISPAC-3 trial, we know that it's not the time of delivery, but it's just the delivery of the expected chemotherapy that makes a difference. But here, Chris Wolfgang group showed that if you have a transitional mesenchymal type and you have a delayed initiation of adjuvant chemotherapy, your survival is much worse. Even if you have a transitional mesenchymal type and you receive the expected chemotherapy within 8 weeks, that effect can be mitigated. This is one of the first examples of how this information can be utilized. Obviously, transitional cells have more of a stem-like and more stemness to that, and there is a lot more to be learned from these cells. Similar information, as I said before, I'm not going to go into the detail. We had a talk about KRAS earlier, but ctDNA is very accurate to predict early recurrence. We have an accuracy between 80 and 90%. The issue with ctDNA, it's hard to capture. My personal interest is in extracellular vesicle, and my research is in exosomal analysis. Why extracellular vesicle? They contain a lot of information from the original cancer cell, and the information contained within the exosome is preserved because it's preserved within a lipid bilayer. So your DNA chiral basis is longer, the RNA is better preserved, but you also have metabolites, protein, and other cellular component. So when you compare this to ctDNA, there are several advantages. One, extracellular vesicle come from live cell. ctDNA usually is from all cell in the body, but when you have a cancer cell, the component of ctDNA can be 50% depending on stages. But it starts to capture, it degrades very quickly, it starts to preserve, and only allows you a short analysis of the target DNA. With my collaborator at CMU, we have developed this mechanism of capture of exosome. This is a lipid nanoprobe with nanoparticles. Without going into much detail, but we prefer this method because it's very easy to perform. And when you compare to a commercially available kit, even with ultrafiltration, has a much higher affinity of capture. With this lipid nanoprobe, we have initiated our evaluation for newly diagnosed pancreatic cancer. In this first phase, we are analyzing exosomal DNA, focusing on KRAS, but the project is to include microRNA. We have 3 or 4 targets for that, and then proteomic analysis. This research in the initial phase was funded by the National Pancreas Foundation. We haven't analyzed so far in the last year and a half, 52 treatment-naive patients. We have about 14 months of follow-up, 35 resected patients have at least a time point following resection. And when I give you the aggregate data, it gets a bit over my head because it becomes exponentially more complex depending on what time point you start choosing. But for the purpose of this talk, I would like to focus on this discordant group. To me, this is the most interesting. It's a group that has response by CA19-9, but has a completely different response by exosomal DNA, or vice versa. When they are concordant, everything is good. But as CA19-9 goes, well, DNA is not captured or decreases, life is good for us and for the patient, but the discordant are quite different. And I'll show you a few patient-level data example here. This is a patient with a borderline resectable pancreatic cancer, was treated with neoadjuvant chemotherapy, received 3 months of FOLFIRINOX, had a CA19-9 that was favorable, like below that 500, had a good response by biochemical, met our criteria of 50% decrease in normalization, and then we did the operation. Operation showed a T2, N2, PNI, LVI positive, a lymph node ratio 30%, and a CAP score of 3 and a positive margin. Now, it could be technical that is positive margin, but usually in this situation is infiltration of the cancer along the lymphatics. You cannot clear their margin. When you look at the exosomal DNA, we started at time of diagnosis with a 0, which means we did not detect any mutation in the exosome of this patient. Now you fast forward this second time point that at the time of the initial study was the day of surgery on the OR table after induction of anesthesia. On that time point, we have now an increase of about 7%. What happened to this patient? He recurred in 6 months, a liver and subcutaneous tissue. Now, subcutaneous metastases are pretty rare. We ended up harvesting this patient for TIL, but eventually he died. And we had this operation before, would have made a difference? We don't know. Possibly. A second example, KRAS G12D, most common mutation, as we heard, CA19-9 now very high, 2,500, normalized. PAT, T1C, N0, LVI, R0, no lymph node ratio, CAP2, so moderate response. What happened to the exosomal DNA? We detected, which in and of itself, it's a bad prognostic sign, and the amount increased now to 3%. Patient died in 9 months. I know we're running out of time, so this prospective evaluation, if you normalize, obviously you survive longer, 36 months in this patient with normalization of exosomal DNA, and then 21 months in this patient that had undetected exosomal DNA. Follow-up is still ongoing. This is what we plan to do with that study, and I don't want to take too much time here, but the reality is this is what we're looking for surgical biomarker. Obviously, optimal biomarker is early detection. If you can prevent, that's ideal. But for surgeon, what's important is detecting patients that are candidate for resection, who is responding to chemotherapy, who is having chemotherapy that is not useful, and obviously detect early recurrence, as we hopefully will have better treatment going forward. Thank you.
Thank you, Dr. Panicia. Would you mind joining us in the panel? Our next and last speaker of this session is Dr. Fon. He's a surgery fellow at the Brigham and Women's Hospital. Dr. Fon, the stage is yours.
Thank you. And this talk is a little bit different from the previous few talks, which were general reviews of very important topics. This was a study that was presented at the Pancreas Club on a single study with original data. So my name is Evan Fong. I'm representing our group from Mass General, and I'd like to thank the Pancreas Fest Program Committee for giving us the opportunity to share our findings on the significance of unstenotic duct dilatation in evaluating IPMNs for their risk of malignancy. So an important area of pancreas research is understanding which IPMNs warrant surgical resection. That is, which IPMNs harbor the greatest risk for developing high-grade dysplasia or invasive cancer, which is what I will refer to when I use the term malignancy. The Fukuoka consensus criteria summarize many of the known risk factors used to stratify lesions based on their imaging features, but perhaps the most salient risk factor is main duct dilatation, which is a harbinger of a main duct IPMN, increases the risk of malignancy by over 5-fold. So in contrast to main duct dilatation, unstenotic duct dilatation has not received as much attention. Although the unstenotic duct is considered a branch duct, it actually originates as a primary duct of the ventral bud, which fuses during gestation with the dorsal bud to form the mature pancreas. Therefore, it's been hypothesized that the unstenotic duct dilatation is also associated with malignancy, just like main duct dilatation is. 2 prior studies from Memorial Sloan Kettering and our colleagues here at the University of Pittsburgh have alluded to a potential association between unstenotic duct dilatation and an increased risk for malignancy. So we wonder if we will find a similar association in a larger cohort at Mass General Hospital. So we performed a retrospective core study of patients who underwent surgical resection for IPMNs, and in total, 467 patients met inclusion criteria, which included having at least one Fukuoka risk factor. Imaging data was collected by HPV radiologists who were blinded to the patient's pathology results. As in previous studies, we define unstenotic duct dilatation as more or equals to 4 millimeter in size. We use multivariable logistic regression to analyze the association between unstenotic duct dilatation and malignancy, both across the entire cohort and just among a subset of patients without any cysts in the head of the pancreas. These are patients who had branched out IPMNs in the pancreatic neck, body, or tail only. In our cohort, the mean age at surgery was 70 years, and 49% of patients were females. Across the cohort, 267, which is 57% of patients, had only Fukuoka variant features, while the remaining 200, which is 43%, had at least one high-risk feature. Unstenoid duct dilatation was present in 35% of patients, of whom 73% of them had malignancy. In fact, this bar graph displays the prevalence of malignancy in different subgroups of patients, each then further divided into those with or without unstenoid duct dilatation. What I want to draw attention to is the finding that the prevalence of malignancy in all patients with unstenoid duct dilatation was 73%, which was similar to the 74% in patients with at least one high-risk stigmata and 73% in patients with main duct IPMNs. To test whether unstenoid duct dilatation was associated with malignancy, we constructed a multivariable logistic regression that adjusted for the presence of at least one Fukuoka high-risk stigmata, thus stratifying them out from those with only worrisome features. And even after this adjustment, the presence of unstenoid duct dilatation was associated with a 2.8-fold higher odds of malignancy. We plotted a version of this regression model here to demonstrate that our model was well calibrated to the raw data. The data series on top shows the odds for malignancy as the size of the unstenoid duct increases in patients who have already had high-risk stigmatas. The data series below is for patients with worrisome features only. In both series, unstenoid duct dilatation is increased risk for malignancy. Next, we wondered whether this association would still hold if we looked at patients without any evidence of cystic lesions in the head of the pancreas. This analysis was inspired by previous literature that had shown such an association in this kind of subset of patients as well. And among 194 patients with branched duct IPMNs in the neck, body, or tail only, we constructed a similar logistic regression in which we again adjusted for the presence of known Fukuoka high-risk stigmata. We found that unstenoid duct dilatation increased odds of malignancy by 3.2-fold in this subcohort. This was an interesting result because it suggests that unstenoid ductal dilatation somehow predicts malignancy even when the only cysts the patient had were far away from the unstenoid process. Returning to the entire cohort and testing association between unstenoid duct dilatation and malignancy in patients who had the head of the pancreas resected versus those who did not, in both subgroups, unstenoid duct dilatation also predicted malignancy. All these findings suggest that whatever process is going on in the unstenoid duct seems to relate to the development of malignancy in the rest of the pancreas. It's hard to explain how a dilated unstenoid duct could be associated with cancer elsewhere in the pancreas, but we speculate that this could be explained by the field cancerization effect, which posits that the development of malignancy in IPMNs is multifactorial and due to an overall elevated risk of cancer throughout the entire gland of the pancreas. So future studies with more granular pathologic findings and molecular genetic studies are needed to understand what unstenoid duct dilatation actually means and its relationship with malignancy detected across the entire pancreas. So to summarize, unstenoid duct dilatation predicts additional risk for malignancy that is not accounted for by current Fukuoka criteria. Patients with unstenoid duct dilatation in the setting of other worrisome features appear to have a similar risk for malignancy as a main duct IPMN or a Fukuoka high-risk IPMN. Hence, we observe that in the presence of other risk factors, unstenoid duct dilatation may be considered a high-risk feature that warrants surgical resection. But nevertheless, the study cannot tell us what the significance of an isolated finding of unstenoid duct dilatation is in Fukuoka negative patients, since all our patients in our cohort were resected patients and not patients who are undergoing surveillance. Thank you so much for your attention.
Thank you, Dr. Font. Please join us for a panel discussion. And we have a few minutes for questions. Dr. Hughes.
Good morning, everybody. Steve Hughes from Florida. First question for our last speaker. Wonderful presentation and congratulations on being chosen as the best of the best from Pancreas Club to be presented here. Quick question for you. What's the magic number for the unstenoid duct size? So it's not a centimeter, right? But it looked to me like it's in the 3 or 4 millimeter range.
Thanks for the question, Dr. Hughes. And that's a really good question. And we did look into it and we did include it as a some analysis in the manuscript as well, because one of the reviewers did bring it up. It seems like based on the literature, it ranges somewhere between 3.5 and 4.5 millimeter is the magic number if you assume that you value sensitivity and specificity equally. And in our study, our magic number was 4, which is reassuringly similar to what's been reported in the literature. So I think that number is 4, but also just to keep in mind that every study on unstenotic ductal dilatation in the literature has been based on patients who have had an operation for one reason or another. And we still don't know what the natural history is and what it means if we see a patient in clinic with an unstenotic ductal dilatation in the context of the denominator of the overall cohort of patients we see in clinic.
Dr. Whitcomb.
Thank you, Dave Whitcomb from University of Pittsburgh. This question is going to begin with Dr. Lucas, but it's going to metastasize to the first session. I loved your presentation. It was very thoughtful and very needed. The question is about diabetes. It turns out that as we're studying diabetes and the pancreas, pancreatic cancer risk seems to be much higher if diabetes occurs after acute pancreatitis rather than independent. And if you segregate them, the risk of pancreatic cancer in diabetics in general is a little bit higher than you'd expect. But if it's post-acute pancreatitis development of diabetes, which is often type 2 diabetes, turns out, is really where most of the risk comes from. With that background, assuming it's true, you've shown a number of risk factors for pancreatic cancer, and I'm wondering what is the necessity of injury and inflammation, such as overt or subclinical pancreatitis, to actually initiate the process of rapid development of pancreatic cancer, because the previous discussions, especially around KRAS and other things, suggest you have to have injury and inflammation to start the process. And so my question for the group is whether or not this risk stratification is like the brakes on your car. You'll never know they don't work unless the car is driving, and you need drivers before the risk factors become important. So your thoughts on that, and maybe a couple others could comment.
Yeah, thank you for that question, and which I think perhaps already did metastasize to the first panel. These types of studies, though a larger epidemiology study, it's really challenging to elucidate causality. We can look at associations, but causality is a real challenge. And we can adjust for that in various different methods. But I think you hit the nail on the head without getting into the mechanism. When we looked at that last study that extrapolated data from the Danish study as well, the Danish cohort as well as the VA study, you see pancreatitis and you see type 2 diabetes pull out. And I almost hesitated because of what you're describing is just using type 2 diabetes as in that setting a risk factor for. Those types of data sets can perhaps help us answer those questions because they're time stamped, right? So they didn't tell us in that study, to my knowledge, we can look at the supplemental tables about the order and the sequence of events between pancreatitis and diabetes. We could go back and look at that to see if they have those data available. But I think that would be a really interesting hypothesis and something perhaps with these very large data sets that we could evaluate. I don't know if it's kosher to have the other comps. Yeah.
Is pancreatitis injury to pancreas necessary to develop pancreatic cancer? Yes. If we have enough, so.
Necessarily have to be injury, just the setting of injury, classification, or current injury.
Good morning. I really like the presentation on early detection and risk assessment. I guess this is one of the few topics that in 2 days of this meeting. I wanted to engage the panels, not just one person. How do you see the role of single organ detection versus multi-cancer detection test? In a sense that pancreatic cancer being such a low incidence disease. And if you look into the history of MSET, multi-cancer detection test, it is really helping to detect some of those which are rare or low incident, but not necessarily with the high sensitivity.
Yeah, thank you for that question. I took all the biomarker slides out of the talk, knowing that you were following. I think it's challenging looking at multi-cancer detection tests at this time because we're having a difficult time knowing where the signal is coming from. When we look at some of the multi-cancer tests that are either in development or on the market, the test characteristics for pancreatic cancer in particular for early stage disease really do seem to be suboptimal. And granted, that's in a general population. And so that's a sort of different kettle of fish there from some of the high-risk populations that we more routinely perform surveillance on. The test characteristics for other cancers are better with some of these multi-cancer detection tests. But I really struggle with what to do with positive tests, not just for the multi-cancer early detection, but for even pancreatic cancer specific early detection biomarkers, because we don't really know what's a true false positive over time, and we don't know how to analyze those data over time. I don't know if you have perspective from surgical.
Yeah, I echo your comment. It's a very interesting question. As a surgeon, we're not really in the field of early detection. We find the lesion and then we treat it. But from a surgical standpoint, I mean, we had a very interesting talk by Anne-Marie Lennon O'Brien a few weeks ago here in Pittsburgh. And one of the discussions she had was this early detection test and the one that were developed at Hopkins. She has some interesting data, but for our own experience here, we have done significant amount of work with detection of precancerous lesion directly into the pancreas or in the biliary tree. And just to bring the attention on more narrow focus, I mean, even in those situations, it's still hard to really identify what's cancer, what just simple inflammation. And the tests are pretty good, they up to 80%. So even in the situation when you have a lesion, you have a suspicion, still you cannot commit that it's true cancer or no. So I think we're still a bit far away from multi-cancer detection on the general population. You need to be, less than 1% error to be useful at a population level.
In the meantime, I have a question for Dr. Fong as a follow-up of the question we had before. When you look at your study, who was doing the measurements of the pancreatic duct and what was the inter-observer agreement with this? Because there can be variability in measuring the pancreatic duct among different radiologists and among different centers.
That's a really good question, and this was a labor of love. But basically, it was our chief of HPD radiology imaging section who was doing it with a fellow of his. So there were two board certified radiologists who were reviewing every single CT scan for every patient in the cohort, blinded to the pathology. Their agreement was, it approached 85%, and they usually would meet to adjudicate any disagreements. So we thought that was the only way we could, in good conscience, do this study, because I don't think it would be responsible for us to be either having a, no offense, but a medical student or a junior resident go through every CT scan and assume that they can measure it accurately. So we had to make sure that a radiologist, a board certified radiologist, two of them with experience in looking at IPMN related cross-sectional imaging, do the measurements. But since my talk was on IPMNs and we were discussing the topic of surveillance at the population level, I don't have anything more positive or optimistic to add to the discourse, but more, not to sound like a pessimist. I think when we're talking about early detection and surveillance, we thought when we started discovering IPMNs that they were it. They were our opportunity to intersect and intervene before they degenerated to cancer. But there are recent data that has looked at the past 30 years, and we have made no progress whatsoever in pancreatic cancer-related mortality in the past 30 years. So whether it's we were better with systemic therapy, whether we had better understanding of the sequencing of our treatments, and whether we were actually intervening sooner with picking up and operating on more and more IPMNs as an epidemic, we haven't moved the needle all that much in pancreatic cancer mortality. So I think it's important for a meeting like this to have all the brightest minds in the room continue work on this. Sorry to be a Debbie Downer.
Yeah, one question for Dr. Lucas, and follow up on, risk factors, if you can make maybe more precise use of what you already have from the data. I was wondering about gender differences, if you would actually have the GWAS cohorts and the cohorts looked at by gender, and also ethnicity, whether you'll be able to identify subsets with already existing models, which provide a little bit more power to predictive risk for, some of these risk factors.
Interesting, the gender question. I hadn't particularly taken a deep dive into that, but I think in consideration of the slides of differences in trends and annual percentage change based on gender, I think that would be really interesting to look at. And that paper actually proposed that it was either some sort of known or unknown environmental exposure, which I think perhaps would make sense given that there's a difference in terms of sort of the birth cohort or the age of individuals. Although when you also look at the PRS score, that did also function differently according to age. So it would be interesting to take a bit of a deeper dive there and examine more closely those interactions. As far as the race question, I think when I looked at the PRS scores that have been published thus far in pancreatic cancer, the only one that really examined a multi-ethnic or diverse patient population was the one that I had shown. And I didn't go through the data there, but that is one of the limitations for polygenic risk, not just for pancreatic cancer, but in a number of other cancers, is that if our prospective cohorts that we're following, and these big cohorts are all European ancestry, it becomes a real challenge to generate PRS for other populations. So there are efforts in that direction. My institution has something called the Mount Sinai Million, which we are aggressively recruiting 1 million Mount Sinai patients within the New York City area. The predecessor to Mount Sinai Million, which is our BioMe program, did have genetic ancestry data, and we have a much more genetically diverse patient cohort, which is expected in New York City. And that Mount Sinai Million will be linked to not just the genetic genomic data, but also is linked to our electronic health record. So we don't have the power of the 6 million Danish patients, but we do have a large, hopefully prospective cohort, which will have those types of data available in a more diverse population. So we really do need, in order to move that PRS forward so that it's applicable to our United States population, we really do need more diverse patients to be incorporated. So thank you for that question. I really do appreciate that.
Dr. Paniccia, the question I have is, when you look at EVs versus ctDNA, do we know which one has more genetic information? And also what differences exist in the type of genetic information you get from both?
And that's a really good question. Thank you. So they are fundamentally different. ctDNA is really a strand of DNA. They usually a few tens, thousands of base. And exosomal vesicle, they are a packet of complete information, everything that's inside a cell. So it has protein, has DNA, has RNA, and metabolites for that matter. For the information that you receive from that, from a ctDNA, you had to look for the information that you want. So you had to develop a primer that captured the DNA of your interest. So it's usually a tumor-informed test when you do it in the setting of response to neoadjuvant chemotherapy. Level for exosome or extracellular vesicle, you can extract all DNA and then do a multi-array search, or you can search for protein or everything else that you're interested, which is within the exosomal vesicle. They really are fundamentally different. Exosomal vesicle come from living cell. All cell in the body release exosome and release mostly all content that is within the cell that is of interest for biomarker, at least for our type of interest for response and prognosis. ctDNA just shedding of DNA of the cell that you go look for it, but nothing else than DNA.
So I got to follow on that with something we don't often talk about, but because I think on the surgery side, you know, it's not quite as impactful to us. But now that we're coming out with more biomarkers, do you think it's important that we differentiate a primary pancreas tumor from a primary distal cholangio? I know we don't talk about it a lot, but our med-onc colleagues certainly perseverate on it. Or do you think the biology is similar enough that it just doesn't really matter?
Yeah, thanks for your question. Interesting question. It depends of your therapeutic approach, right? For distal cholangio, currently there is no recommendation for neoadjuvant chemo. Not that in pancreas there is suggestion, there is no definitive recommendation, but definitely some stronger study. For distal cholangio is not the case. So now the dogma is to proceed with resection. Biologically, they both pretty bad. So I don't know if they really will make a difference, know if it's a pancreas or distal cholangio, because the therapeutic efficacy of what we have now is suboptimal. I think as you move forward, yes, it will become important as we start developing more targeted therapy, more efficacious chemotherapy that differentiate in efficacy, then that will be fundamental. We do go through an exercise every time we have a distal ampullary lesion in the distal bile duct or head of the pancreas to differentiate. You have about a 20% error rate as things stand for recent reviews. Currently, I'm not sure it makes a big difference unless you are a true proponent of neoadjuvant chemotherapy, which we are. So in every case that we suspect a pancreatic cancer, we will give chemotherapy. So for us, makes a difference, but biologically, I don't think so. And genetically, they are not that different, at least for the biomarker that we have now through sequencing, the brushing, or the biopsy of the bile epithelium of the pancreas. They are hard to differentiate. In fact, when you have molecular biomarker, the question always goes back to the gastroenterology. Is there a mass in the pancreas or just a stricture in the bile duct? That's how you know which one it is.
I have one more question for Dr. Lucas. After we detect the risk factors and we have high risk patients in our cohort, let's say that we did biomarkers and the biomarkers are consistent, that there may be a malignancy. How do we actually do imaging? How do we stratify the frequency of follow-up, what type of imaging, endoscopic ultrasound versus MRI, and how frequent to do it?
Great question. You know, I live in New York City and work in New York City. And so we have several large academic centers that have high-risk surveillance programs for individuals with strong family history or with pathogenic germline variants in pancreatic cancer susceptibility genes. And if you go to different institutions, which are within walking distance of each other, you will get different answers. And the same is true across the country and worldwide where those surveillance programs exist. You know, we have general consensus guidance. The CAPS consortium has published some, the American College of Gastroenterology, ASCO has provisional guidance, AGA has some guidance. Our ASGE, our endoscopy colleagues have published guidance, which is, you know, slightly different. And it all sort of centers around largely annual surveillance with either endoscopic ultrasound or MRI. And there are some centers that are proponents of a EUS-based approach. There are subcenters that are proponents of an MRCP approach. Some centers that alternate between the two. Certainly different centers, I think, have different strengths. And so if your endoscopic program is very strong in one center, you might decide one or the other. And then if the one test is giving you more information, then sometimes I think that's the useful test to pursue in particular individuals. So for example, I have some Lynch syndrome patients with incomplete and complete intestinal metaplasia and a family history of pancreatic cancer. In those individuals, I might skew towards endoscopic ultrasound, but typically without any worrisome or concerning features, annual surveillance and the method TBD. But these are all research questions that are important to move the field forward.
If there are no more questions, I think the session is over. Thank you so much to the panelists and to the speakers.